All right, so microscopy and cells. So when you're talking about microscopy, now when these cells were discovered, it was necessary to know a microscope because it enabled those people who discovered these cells to be able to view what they are talking about. Okay, so cells themselves were discovered by Robert Hooke, that guy. Let me write his name. Robert Hooke. And these cells were discovered in the year 1665. So a microscope was necessary in order for us to be able to view the cells which were discovered by this person. Now, a microscope is basically an instrument which is going to be used to make small objects big. As we know, cells are we describe cells as microscopic. And the meaning of that is that they are too small for our naked eyes to be able to see like that. So you need something that will make them big for you to be able to see. Now, microscopes themselves, they are of different types. So light microscopes, we can distribute them or divide them into either being optical microscopes or electron microscopes. Optical, every time you hear of optical, it has to do with light. So optical microscopes, these are microscopes which use light. So you want to view things, but then you have to use light for you to be able to view those things. Electron microscopes are going to utilize, or they are going to use electrons for you to be able to see those things that you want to see. Now, the optical microscopes are of two types. They can either be single or simple microscopes or compound. That's from the word simple means you're only using one lens. A lens is that part that you use to make things big. Compound microscope uses two lenses. So the lenses here, you have got the objective lenses and the eyepiece. So the eyepiece where you're going to place your eye also has got lenses. Yeah. So these are what we are talking about. You can see for a simple microscope, you only have where to view from here. Okay, where to view from, and then you get to see your object. So it has no objective lens, but it has got a magnifying lens. A compound microscope, this is the one that you use. It has got the eyepiece and also the objective lens. The eyepiece will be here where you are looking, and then the objective lens, these are the ones here where you can actually be changing those objective lenses. If you want to go a bit in details, those compound microscopes can also be of different categories. You can have bright field microscopes, dark field microscopes, phase contrast, fluorescent, polarized. If you hear of bright field, this is a microscope. When you are looking at something, the background, you can see my board. My board is white here. So if I use a black marker, you can see. Now imagine that the board was black. Okay. So when the board is black and then you want to view things, it means the things that you should be able to view should be a bit of whitish, right? So such a microscope where the background is dark is called dark field. A bright field microscope is a microscope where your background is white or some kind of light. Okay, so that is those are the ones, bright and dark. Bright background is bright, it's white. Dark, opposite. Now I don't want to describe these more in details because they are my I don't think they are of your level. Okay. So it's important to understand the parts of a microscope and also just their general functions. A compound microscope we are describing here is going to have the eyepiece where you are going to be able to place your eye. So the eyepiece is going to be also necessary in making sure that the object is magnified, the object becomes big so that you can be able to see it. You also have the body, okay, you also have the body there of the microscope. You also have the limb. So I can also describe this as the body in some books. You'll see this is used, this part known as the limb is going to be used for holding the microscope. Also the base. So you place your hand down on the base and then you also hold the limb. And then you can be able to move your microscope. You have the adjustment knobs. You have got the coarse adjustment knob and the fine adjustment knob. I'll describe this more. You also have the revolving nose piece. So this is the nose piece here. The nose piece holds your objective lenses. Your objective lens is there to further magnify, to further make big your object. So magnification is just making big the object. So magnification is going to be given as the magnification of the eyepiece. Let's say the eyepiece is magnifying things 10 times, and then the objective lens is magnifying things 40 times. So my total magnification is going to be 10 times 40, and so the total magnification is going to be 400 times. So we put that x to represent the times. Now magnification can also be described as how big something has become. So magnification is going to be size of the image over size of the object. The image is what you are able to see. Let's say when you put something on the microscope, when you look at it, it is looking as though it is, let's say, 5 times the thing that you have put. Okay, let's say you have put something on the microscope. When you look at it, the length is looking as though it is 10 centimeters. But the actual thing, so that is known as the image, that thing that you are seeing on the microscope. But the actual thing that you had put on the microscope, after you measure its length, you find that its length is just 2 centimeters. So magnification is image over object, so that is going to be, so this is the object, the actual thing that you had put on your stage. So the stage is where you place something that you want to view. It will be 10 centimeters over 2 centimeters. You can see the units cancel out. So magnification is unitless because this will cancel out. So it will be 5. So magnification is 5 times. So you just say 5x. Okay. And then you are also going to have a mirror here. So the work of the mirror is that when light comes, light actually bounces, it goes there. Remember we said these are optical microscopes. They use light. And light will be necessary for you to be able to view what is here. Without that light, you will not be able to see what is there. You also have the diaphragm, which is down there, and also a condenser. So the parts of a microscope, you can divide them into two parts. You have got mechanical parts and optical parts. Optical parts, these are parts which are going to involve light passing through. Mechanical parts, these are parts which you do other functions, not other than allowing light to pass through them. So for the optical parts, we have got the condenser. The condenser is going to converge the parallel light rays as I described them here. You can see the rays were coming like this, they bounce and then get to go like that. So that is converging the light rays. Okay. Yes. Mm-hmm. Mm-hmm. So that would depend with the information you'll be given. If they've given you the magnification of the eyepiece and the magnification of the objective lens, just multiply the two. But if they've given you the size of the object and the image, divide. Is that good? Okay. If they've given you the magnification of the objective lens and the magnification of the eyepiece, multiply them to get the total magnification. If they've given you the sizes for the object and the image, divide image over object. But you always make sure that the unit of the image and object is the same. Otherwise, you are going to find something else. Is that good? Okay. Another optical part is the objective lens. Objective lens is basically to magnify objects. Same with the eyepiece. To magnify your object as you get to place your eye there. These other functions we had described them. What I didn't describe is the coarse adjustment knob and the fine adjustment knob. The coarse adjustment knob, these are also known as screws because like you'll be turning them. So the coarse is for bringing the section into focus, while the fine is for focusing the section with high power objective. So the thing is this, for the coarse adjustment knob, when you move this thing, when you move this, what will be happening is that you are actually moving the entire of this thing either down or up. And as you are moving that thing either up or down, you want to make sure that you find a position that is going to be okay for you to clearly view what you have placed on your stage. Now, for the fine adjustment knob, you are moving it to make sure that your object becomes clear enough. Okay? If it was looking a bit of blurred, you are adjusting it so that it can look more clear. So they are all being involved in making sure that you bring your image, let's say the specimen, into focus. You want to make sure that you can see it clearly. So the course for bringing the section into focus with low power objective. So one is just describing more. The what are these? The objective lenses. They are supposed to be, most of the microscopes you will see, they are going to have three objective lenses, which you'll be able to turn. So you can actually be rotating this thing which holds the objective lenses is the revolving nose piece. Not like that because you can actually rotate it. This can be high power, this one medium power, this is low power. So when something is at low power, it means that you can't see very much details, something that you've placed on your microscope. Let's say you are viewing a cell. There are some things that you cannot view at low power, okay, unless you increase the power. So when you want to view something with at low power, you want to focus it, you can adjust a coarse adjustment knob. Remember we said the coarse adjustment knob is just to move your entire barrel or the whole of this portion, this structure here, up or down. So it means that you will not be able to view something much in detail. But at high power, if you want to see something much in detail, you can now focus it using the fine adjustment knob. You are rotating it so that you can see pictures more clearly. Okay. You also have your mirror. Oh, here I was describing the function of the mirror for the condenser. The mirror is the one which will direct the light rays. Now the condenser will be to converge the parallel light rays. Here is the condenser. I was describing something else. The condenser is at this point. Look at where it is here. So let's say the light rays from the mirror, there are a lot of them. Going like that, going like that, going like that. You want to converge them so that they all come at one point. They all come at one point. That will be the function of the condenser, bringing the light rays at one point. Okay, that is to converge them. Okay, so that is basically, those are some things that you want to look at on a microscope. Now, when you are viewing things on a microscope, let's say you have got a very tiny object, very tiny objects. When you are viewing them at a low power, you might view them as one thing, okay? But then as you get to increase the power, you notice that those are actually two different things. So the the minimum distance at which you are able to see two different objects as separate, that is known as, uh, what's that term again? It has gone. Anyway, let me remember the term so that I describe to you. Let me mention this again. When you are at low power, two objects might be seen as one. Think of it. When you are very, very, very far, let's say you are standing here, and then you are looking at people who are very, very far. Okay, let me not just say people. Let's say you are looking at an object which is very far. You might see as though it is one object, but as you get to go near that object, you actually see that those are two different objects. Okay, two different objects. So that is what I'm describing. I'll tell you the term if it comes back. But remember that. Okay. Now let's describe cells. You know now how to use your microscope, but now you want to identify the contents of a cell. You want to see the parts of a cell. The first thing you need to understand is that cells are of different types. And what you want to understand for you at your level is you want to differentiate cells. Some cells are eukaryotic, other cells are prokaryotic. Okay? So you have got eukaryotic cells, and then you have got prokaryotic cells. What is important is for you to understand the difference between these two types of cells. One thing is that eukaryotic cells have got membrane bound organelles. Anyone, what is an organelle? What is an organelle? In any language or however you can describe it. What is an organelle? A living organism. Okay. Any other try? Any other? An organelle. It's the same thing as you did in high school if you did. Oh, it just seems we didn't do. Okay. All right. Let me first tell you about what I was describing. The minimum distance at which you can see things are separate, that is known as a resolving power. Resolving power of a microscope. An organelle is a structure or a part that is found within a cell. Okay? If you have got a cell, in a cell you are going to find things like a nucleus. These are known as organelles. Organelles. Okay. Now, we are saying that for eukaryotic cells, these organelles, these things that you find in a cell, they are going to have a membrane. Just like the cell itself has got this structure surrounding it. That is what we call a membrane. Same with these structures that you are going to find inside the cell, they are also going to have membranes as the things which surround them. Now, for prokaryotic cells, they are going to have structures. Okay, okay. So basically, a structure found within a cell and it is bounded by a membrane, right? So, in simple terms, so a membrane is just like a cell itself. If it didn't, it's more like a cover, okay? Without this thing which I've written in black, it means that things which are inside will just break off, all right? So, that is a membrane. Same, those parts which are found in eukaryotic cells inside the cell, they are going to have a membrane. Prokaryotic cells do not have membrane bound organelles. They can have some things found inside, but they are not going to have membranes. They will not have membranes. I'll describe a prokaryotic cell to you. The other thing is that a eukaryotic cell has got a nucleus. Prokaryotic cell has no nucleus. This is one important difference between a prokaryotic cell and a eukaryotic cell. Every time you are differentiating this, you are going to be able to mention that. Okay. Now remember from your high school, the nucleus contains DNA. What you determine the characteristics which you have is actually DNA, deoxyribonucleic acid. That's what we call DNA. So meaning for prokaryotic cells, since they do not have a nucleus, it means that the DNA will not be in the nucleus. Okay? Means that the DNA will just be in the cytoplasm. So these are some big differences that you want to describe. Yes, Michael? Was that a hand? Okay. Now let me describe for you more on a eukaryotic cell before we can talk about a prokaryotic cell. Now eukaryotic cells of our interests we are going to discuss is eukaryotes are, are basically either animals or plants. Eukaryotic cells is describing those cells which have got a nucleus and membrane bound organelles. Simplest examples we describe are animals and plants. So you need to know an animal cell and a plant cell because these are examples of eukaryotic cells. Okay. Now for an animal cell does not have a definite structure. So you just see it like that. It's not definite. It is irregular. These are some differences. Now for a plant cell is going to be regular. This one is going to be regular. Another thing is talking about the nucleus. An animal cell is going to have a nucleus, and the nucleus of an animal cell will be mostly on the periphery, meaning it is not on the center. Okay, the nucleus is on the periphery, meaning not on the center. Now, for here, I've changed this. This thing, the nucleus will be the one on the center, centrally placed nucleus. And then a plant cell, the nucleus will be the one on the periphery. Okay. Can also say peripheral nucleus, meaning the nucleus is not on the center. A plant cell is also going to have a vacuole like that to actually cover the almost the biggest part of the cell, the vacuole there. Now for an animal cell, it can or cannot have a vacuole, but mostly you say it has no vacuole. A plant cell has that vacuole. Another important thing is that a plant cell is going to have two membranes. The outermost is the cell membrane, and then the one which is inside, the outermost is a cell wall. The one which is inside is a cell membrane. Now, okay, let me first label this so that you see this is the vacuole, this is the nucleus. These are not the only parts which a plant cell and an animal cell is going to have, but the common parts which these two cells are going to have is the animal cell also has a nucleus. It So this space between the vacuole and the cell membrane is the cytoplasm. The cytoplasm is going to have two things. It's going to have the cytosol. It is also going to have organelles. Okay. The cytosol is the liquid part of the cytoplasm. The organelles, these are structures that you're going to be able to find in that liquid. We have got things like the nucleus. We also are going to have ribosomes. Okay, you are going to have ribosomes. These are small things like that. Ribosomes. Apart from that, you're also going to have different other structures like what else? Chlorophyll for a plant cell. Okay, chlorophyll is the one which makes the green pigment. Now, what makes chlorophyll are the chloroplasts. So we're going to have chloroplasts to be able to make the chlorophyll. In animal cells, you also find things like the endoplasmic reticulum. This is a structure that you are going to see like something like this. This will be the endoplasmic reticulum. Now, the endoplasmic reticulum can have ribosomes on them. So, if you see dots on the endoplasmic reticulum, it will be known as the rough endoplasmic reticulum. Rough because of the presence of those ribosomes. If it does not have ribosomes, then it is known as smooth endoplasmic reticulum. You also see organelles which are like this. They are more cylindrical. These are known as the mitochondria. The mitochondria is used to make energy. So it's called as a powerhouse of the cell. It makes energy for the cell. The energy is made in forms of ATP. ATP is known as the currency. Currency, is money used to buy things, right? So ATP is known as the energy currency. It is the form in which we use energy in the body. You are going to have many other different structures like lysosomes. You are going to have a lot of other things. So these are some of the organelles. But the thing is that for animal cells, their outermost layer is a cell membrane. It doesn't have a cell wall. So the cell membrane allows things to go in and out of the cell. Things can be allowed to go in and out of the cell. So not everything can enter the cell and not everything can leave the cell. Only those things which are small enough to be able to pass through the cell membrane can actually go inside. That will be the same function of the cell membrane in the plant cell. Now the cell wall is used for protecting this plant cell from bursting after it gains a lot of water. So if this plant cell gains a lot of water, it will not be able to burst. But when an animal cell gains a lot and a lot of water, it will burst. And so it undergoes what is known as cell lysis. Okay? Cell lysis. But when it loses water, it will become very small, and that is going to be known as crenation. Is that okay? So when a when a cell when an animal cell lose, loses water, when an animal cell loses water, let me describe that more. Say we get this cell, okay, we make it lose water to become small. You can see how it has become. If it gains water, becomes very big. So when it gains water, it can break. So what you see is it will break like that. Okay, it breaks off. And that is called the cell lysis. Because it gained too much water and then it had to break. When it loses water, it becomes small. That is known as crenation. This is for an animal cell. For a plant cell, remember we have said it has a cell wall, so it can't burst. If it gains water, it will just become bigger. It might even a bit of lose its structure. So it becomes strong. That is called turgidity. When it has it has gained a lot of water, so it becomes very big. But if it loses water, the cell membrane will still remain like that. But inside the cell will still remain like that. But inside the cell membrane becomes small. The cell becomes small inside. You can see the way it has become inside. So this is known as it has plasmolized. The cell has become small because of the loss of water. That is known as plasmolysis. Becomes big because of gain. That is known as turgidity, but it will not break. Okay. What haven't I described the function for? The vacuole contains a fluid which is known as the sap. Okay. It contains a fluid. So the membrane that surrounds the vacuole, this membrane that you can see, which is surrounding this vacuole, is known as a tonoplast. And it is filled with different things, different molecules which will contain enzymes and just a lot of different things. And the aim is that it will be able to maintain pH, remove harmful things from the cell, waste substances, to store proteins and such things. So that will be the function of the vacuole. Okay. The nucleus contains genetic information. Genetic information is contained in DNA. It is the one which will determine how tall you are going to be, how short you will be, how your eye color, and all your characteristics is contained in DNA. How you are really going to be. What other thing here? Mitochondria already said it makes energy. Now the endoplasmic reticulum. The rough endoplasmic reticulum contains these ribosomes. Ribosomes, these small structures which you can see, they are involved in making proteins. They'll make different proteins. You know, your nails are actually proteins. Okay? Or in other words, the entire body is just basically protein. So what are we saying? It is there for growth. So making proteins. Proteins are the ones which make sure that a person grows. So that's the function of the ribosomes. Because the ribosomes are now found on the endoplasmic reticulum, we call it RAF ER. So the RAF ER will be involved in transporting those proteins which have been made. But the smooth ER doesn't have, so we can just write as RAF ER, RER, or smooth ER like that. The smooth endoplasmic reticulum is going to be involved in making lipids. Okay, making lipids and these other things. When you talk about some cells, some parts, some organs like the liver, you are going to have both the smooth and the rough ER. They are going to be involved in removing toxic substances. Okay, removal of toxic substances. Those are some of the functions. I think I've described all the parts here. Okay. Now, those were eukaryotic cells. We had that cell, the cell wall, the cell membrane, and the other parts which we have described. Okay. Now, just something you need to know about the cell wall of a plant cell. This cell wall. The cell wall is made up of a carbohydrate which is known as cellulose. We'll be talking about biomolecules, so we'll talk about carbohydrates. You need to really understand because they are like they'll carry marks in your exam. So just take note, the cell wall is made of the carbohydrate cellulose. It's actually a polysaccharide. Now talking about prokaryotic cells. A prokaryotic cell is a cell that does not have a nucleus. So I'll write it like this. Remember we said it doesn't have nucleus. So since it does not have a nucleus, you expect that the DNA should be found somewhere else. So the DNA will just be found in the cytoplasm. But then a prokaryotic cell is more similar to a plant cell. Just like a plant cell at the cell wall, a prokaryotic cell also has a cell wall. So the outermost part. But before the cell wall for a prokaryotic cell is also going to have a capsule. If you look at a leaf, you just get any leaf, for example, a leaf from a mango tree. It's going to have that shiny outer part. That is what we call a capsule. So inside the capsule now we are going to have our cell wall there. Okay. So there is our cell wall. Inside the cell wall, that's where now we are going to be able to find the cell membrane. There is the cell membrane. Okay, like that. So inside the cell membrane, that's now where we're going to have the cytoplasm. And remember the cytoplasm, we said it has got the cytosol, which is the liquid part, and the organelles. Some organelles we are going to find will include things like ribosomes. Okay, we are going to have some ribosomes there. You are going to have mitochondria, different organelles as we saw also from the previous one. But then DNA here is found in two forms. DNA is not in the nucleus, so we are going to see DNA just together like that. So it is known as a nucleoid. Doesn't have, there is no nucleus. DNA is just found together, and it is known as nucleoid. But you're also going to have other DNA which is found secular. We call it secular DNA, and that is known as the plasmid. Okay, secular DNA. So a prokaryotic cell has got DNA found together, packed together, known as a nucleoid, or found as a plasmid. So these are some important things. Now, the cell wall, just like we described for an example, I didn't even give an example. An example of a prokaryotic cell you can give is bacteria. There are different forms of bacteria. You have got the gram positive, the gram negative. This is just describing how they stain. But the most common bacteria that you talk about is E. coli. This is just a letter E. E. coli. This is a bacteria. It's a prokaryotic cell. The E is representing this. It's just that it's strict to pronounce. That is why people just say E. coli. Escherichia coli. That's supposed to be the name. But most you hear it as E. coli. Okay. So what is the function of the cell wall? The cell wall is there to prevent the cell from bursting when it gets water. And the entry of water, the movement of water from one region to another is known as the osmosis. So it prevents the breaking of the cell by the movement of water by osmosis. Okay, so that is the function of the cell wall. Hope that is okay. Now, one thing that is important that you need to take note about, of course, we have already described that the bacterias. Now we can describe the type of bacteria looking at the cell, the way the cell wall is. Okay, the cell wall is going to be made up of a substance which is known as murein. It's different from the cell wall of a plant cell which was made up of cellulose. It's going to be made up of a substance which is known as murein. Now, murein is made up of a polysaccharide. If you hear of polysaccharide, it means that these are carbohydrate. This is a polysaccharide, and then it's going to have short peptides. If you hear of peptide, peptide is basically a protein. Okay, so this is a group of amino acids. What makes up a protein are amino acids, just like what makes up the entire human being. Because what will happen is that when cells come together, they form a tissue. A lot of tissues come together, they form an organ. A lot of organs come together, will form a system, and then a lot of systems coming together will form an organism. So just like an organism, like a human being comes from cells, proteins also come from amino acids. Okay, like that. So when you look at the cell wall, you can describe a prokaryotic cell as either being gram positive or gram negative, just looking at the cell wall. So this E. coli, which I've described, is actually an example of a gram negative bacteria. Okay, gram negative. And talking about gram negative and gram positive just has to do with when you stain these cells. A stain is basically a dye. When you stain this thing with a dye, think of it, you get this cell, you put it in ink, which is, let's say the ink is blue. If this cell maintains that blue ink, so you say it has retained the ink, so it is positive for that ink. Gram positive bacteria or gram positive cells maintain or retain the gram stain. Gram negative bacteria will not retain. Say, for example, you place the cell in blue ink, but then after you remove it, the blue ink moves away. So that's a meaning. Okay. And then the cell membrane, which is also known as a plasma membrane. This one, as we said, it is selectively permeable. It only allows some things to be able to pass through it. So that is selective permeability. It is also known as partial permeable. Okay. So in some bacteria, the cell membrane is going to be used for photosynthesis, for making in photosynthesis. What do you make? You make energy, right? You are using sunlight and water and those things. Okay. So the cell membrane is going to be the structure of the cell membrane is interesting. It's going to be, so the cell membrane is described as a lipid bilayer. So when you get that cell membrane, you view it on a microscope in detail. It is a lipid bilayer. And what you mean by bilayer, it means that it has got two layers. So the lipids which make up this is known as a phospholipid. So the cell membrane is also known as a phospholipid bilayer because the type of lipid that makes up the cell membrane is a phospholipid. A phospholipid is made up of a phosphate group which are going to be bound. Not really okay. The way, if I want to tell you this in detail, or let me just mention that we have got what we call glycerol. We'll talk about this when we talk about biomolecules. Glycerol is made up, it's a compound like this. It has got OH, OH, OH. So there is a carbon atom here, there's a carbon atom here, there's a carbon atom. So the carbon atoms are bound to OHs, hydroxides, and then the rest of the atoms are hydrogens. This is glycerol. And then you have got what is known as a fatty acid. A fatty acid is a molecule. It will look like this. This zigzag at every end, there is a carbon bonded to hydrogens. Carbon bonded to hydrogens. And then at the end, it has got this group. Double bond with oxygen and then OH. So this group here is known as a carboxylic acid functional group. So this is glycerol and then this is a fatty acid. So this functional group you can see here, OH, is known as an alcohol. So when the alcohol of glycerol reacts with the carboxylic acid functional group, you'll be able to form a new compound. So that new compound is going to look something like this. What will happen is that this OH will go with this hydrogen, so you form water. And then this oxygen that remains will be bonded to where that OH has gone, so this is what you form. So this will be the compound that you are going to have. That will be the same also here, the same thing. Remember there is a double bond with oxygen, same also here. Remember there were three OHs at this point, like that. So the rest of the things will be the same way they've been H, H here, also H here, also H here, and H there. This molecule that we have formed when we get glycerol and the three fatty acids is known as a triglyceride. It is known as tri because there are three. If you only add one fatty acid with one glycerol, it is known as monoglyceride. If we add two fatty acids, this one and this one, diglyceride. This one is known as a triglyceride. Okay, tri because there are three fatty acids. It is also known as glycerin. You know glycerin, the one you put in lotion, okay? Like that. Now, when this triglyceride, you remove one fatty acid which is here, you put a phosphate group. So it becomes a phospholipid. So what you see it is, we can easily write it like this. And then we have got another fatty acid, only two. The third one is a phosphate, and we write it this side. Now, a phosphate is negatively charged. So this part of a phospholipid is known as the head. This part of the phospholipid is known as the tail. Every time you have got carbon bonded to hydrogen, this thing is known as the non-polar. The meaning of that is that it repels water. It can't come together with water. That is why you see cooking oil can't mix with water, because cooking oil is a lipid. So this tail, the tail of the fatty acid, can't mix with water. Because it can't mix with water, it hates water. So it is known as a hydrophobic. Hydrophobic means water hating. This part of the phospholipid known as the head, because it is charged, water is like this. Water is also charged. So water and the head will come together. So the head loves water. So it is known as the hydrophilic. Hydrophilic meaning water loving. The tail is water hating. Okay. So this is what will make up the cell membrane. So let me describe it now more so that you understand what happens. For the cell membrane, we are saying it is a lipid bilayer. So for a phospholipid, we'll describe it easily like this. We'll write this as the head, and then we'll write the tail like this. These are the two fatty acids, and then the head, which is made of the phosphate group. The head is made up of a phosphate group, the tail is made up of fatty acids. The head loves water, the tails hate water. So for the cell membrane, you are going to see it like this. So the heads are facing in one direction, the tails are facing in one direction. You also have again heads like this, the tails are facing towards each other. So the tails face each other, the heads face each other. Okay, so this one I've written is actually going to be the one surrounding the entire cell. If this is the cell membrane, you are going to have the heads inside and then the tails outside like that. Some heads will be pointed outside and then the tails inside. So the heads are exposed to the outside of the cell because the outside of the cell has got water. The heads are also exposed to the inside of the cell because remember inside we said you have got a cytosol which has got water. But the tails, these are in between. Because the cell membrane is a lipid, it means the things which do not dissolve in lipids can't pass through the cell membrane. Okay? Things which love water cannot pass through the cell membrane. So water itself is not supposed to pass through the cell membrane. But because there are some openings in the cell membrane, those are the ones which will allow water to pass through. And because water is small, that will be what will make it to pass through. Otherwise, things which are big cannot pass through. But the things which can dissolve in lipids, for example, oxygen, nitrogen, hydrogen. These are known as nonpolar gases. These can easily pass through the cell membrane. Okay, those which are polar will not be able to pass through. Example of polar gases like carbon dioxide, you don't expect them to just pass through normally. Also, big things cannot pass through the cell membrane because there are only small spaces that can only allow small molecules to be able to pass through. So that is the cell membrane. Most important thing is that it is a lipid bilayer. And this lipid bilayer, what you'll be able to see, what I've described here is I've just enlarged this line we are writing like this. Okay, I've just zoomed in of this thing here. If you zoom in, that's how it's going to look like that. This lipid bilayer is also going to have some other things. It's going to have some proteins on them and also some carbohydrates on them. And those are just important for some other functions. Like for a cell to be able to know that this thing is not harmful, you are going to be able to use these carbohydrates on top. Okay, those carbohydrates on top. Those will be able to identify things which are not harmful to the cell. And so those are known as surface recognitions or signaling molecules. They will send signals. If anything that is wrong has come in contact with the cell, they will send the signal to the entire cell. And that those are some things that will make people to be sick because the body will start reacting because they've seen something that is harmful. Okay. So that is that. Remember, I also talked about plasmid for a prokaryotic cell. This plasmid, these are small pieces of DNA. They are separate from this DNA. This DNA is known as chromosomal DNA. We'll talk about genetics so you understand what a chromosome is, but this one is known as chromosomal DNA. Plasmid DNA is different from chromosomal DNA. Okay. So these ones can also, DNA is responsible for formation of all different proteins in the body. So this one is also can also form some proteins, but it is not essential. It's not necessary for the survival of a bacteria. Now, without chromosomal DNAs, an organism cannot function, cannot survive because it's the one which will make you to form basically everything about your body will be formed because DNA has said form it. Your nails, your legs, your head, everything because DNA has said form it. Okay, so basically that. Ribosomes. Now, ribosomes, I didn't describe them more. Ribosomes themselves are used to make proteins. Now, ribosomes are of two types. You have got the small units and the big units. Depending with whether you're talking about a eukaryotic cell or a prokaryotic cell, they will be different. So S will represent the small units, and then L will represent the large units. For a eukaryotic cell, the small subunits are called 40S. The large subunits are called 60S. For prokaryotic cells, the small ones are called 30S. The large ones are called 70S. We are just moving from 30, add a 20, you go to 70. From 40, add a 20, you go to 60. This was supposed to be, yeah, 60. This is supposed to be 50, sorry. You are adding a 20 to the small to get the next one. So for meaning that in eukaryotic cell, a ribosome, when you combine the small subunit and the large subunit, that will be the one which will form now the combined ribosome. So the ribosome in a eukaryotic cell is known as the 80S. Now this is not a number, it's not 40 plus 60 to form 80S. Okay. So if you have got a small 40s, just add a 20 to give you the large, which is a 60s, add a 20 to give you the 80s. Same with the prokaryotic cells. The small subunits are called the 30s. You add a 20, you get the 50s, which are the large. Add a 20, you get the 70s, which are the combined. So this will be now the ribosomes of a prokaryotic cell. The S is representing what is known as the Sedberg unit or Sedberg constant. When you get a ribosome, you put in a test tube, you close, and then you start rotating this using centrifugation. How much ribosomes will be able to settle at the bottom? That is what is known as the Sedberg unit. Okay, so that is what the S is representing. In a cell, we have got organelles. Remember, we said the cytoplasm has got the cytosol and organelles. Now, we described an organelle as a structure which is found in the cell. It is metabolically active, meaning that it is involved in making things, helping the cell in destroying other things and such. And it also has a membrane. For example, the nucleus has a membrane. But you also have other things which do not have membranes inside the cytosol. So those things which do not have membranes inside the cytosol, they are called the inclusions. Okay, inclusions. Example is that you are going to have some carbohydrates like glycogen. Glycogen is a carbohydrate. It doesn't have any membrane to surround it. So it's an inclusion. Okay. So this is an overview on cells and microscopy.